Hey, have you ever wondered how to make something glow? About mRNA, that molecule which we've probably heard about over the past while. There's awesome news that scientists have found a way to make it glow. What's up with it? Let's keep watching to find out. Making molecules glow is no new thing, but making mRNA, one of the most important ingredients in our cells, and a potential target for therapeutic drugs and vaccines, both glow and work well at the same time is no easy task. mRNA, which comes from the name messenger RNA, is a biomolecule that passes instructions from DNA to the machinery inside cells, telling them what to make and how to operate. By the way, the mRNA-based vaccines used to fight the coronavirus precisely has mRNA that were developed to tell the machinery inside our cells to make materials that simulate the presence of the virus, which are then used to train our immune system to learn, recognize, and fight the virus in case if it enters our body. So back to the topic, what's up with the glowing mRNA and what can it do? mRNA, while already composed of tens of thousands of atoms, is still too small to be seen by a microscope, so scientists typically use indirect methods to understand how it operates inside a cell. However, knowing where it is exactly inside a cell has been a challenging problem. If we can let mRNA glow, then in principle we can use the light we observe to track where it is inside the cell. Recently, a team of researchers from Sweden and France just achieved that. Their research, which was published in the Journal of the American Society, not only provided a new tool for probing biological molecules, specifically nucleic acids, inside cells, but also opened up potential new avenues for research in biochemistry and molecular biology. Research into mRNA-related drugs and vaccines could well benefit from this innovation. So how did the scientists make the mRNA glow? Molecules with certain special structures can be made to glow. This is a phenomenon called fluorescence. How fluorescence works has to do with a lot of super cool quantum mechanics, but that's a topic for another video. mRNA essentially is a huge molecule. So you can imagine that if you take some of those special structures that allow molecules to glow and incorporate them into mRNA, you can in principle make it glow as well, right? So that's what the scientists did. They changed certain parts of the mRNA structure to include molecular parts that can glow. If we zoom in towards mRNA at the molecular level, we can see that it's made out of certain units like building blocks that repeat. These are called nucleotides. The researchers basically asked, can we modify the chemical structure of a certain type of nucleotide to allow it to glow by fluorescence and use that glowing nucleotide as a building block for synthesizing mRNA? If so, can the synthesized mRNA operate like a normal one inside a cell? After many experiments, the answer was yes. Not only could the scientists make mRNA from the glowing nucleotide building block, but this modification to RNA didn't affect it much from operating normally inside a cell. To make things clear, this wasn't the first time that people have modified mRNA to make it glow, but the methods developed previously have many limitations. They either involve large structural modifications to the mRNA, and thereby disturbing its normal operation inside a cell, or when only the two ends of mRNA are modified to minimize the structural impact. The light produced was too weak to be observed inside a cell. These researchers applied a trick that worked. By doing small modifications to the nucleotide building blocks and using them to make mRNA, they not only successfully made the mRNA able to produce enough light from all of the incorporated glowing nucleotides fluorescing together, but also minimized the overall structural changes towards the synthesized mRNA, thereby enabling its normal operation inside a cell. One can then now track the mRNA inside the cells via their glow and get useful information for studying many biological processes. Anyways, I think this is a really cool piece of research which could potentially play a significant role in helping the development of mRNA-related drugs and vaccines. Just like any other scientific study, this one is loaded with details that can't possibly be all explained in a short video like this. What I've done is to try and share the exciting parts that hopefully will make you get a glimpse of what scientists around the world have been doing, and at the same time make understanding rigorous scientific research a bit more accessible. To learn more about this study, I've attached a link to the paper below. Okay, that's it for this video, and thanks for making it to the end. To see more videos like this, remember to hit that subscribe button. Thanks again, have a good one, and see ya!